Hello students, in the previous videos we have seen the introduction for biomedical instrumentation, different types of biomedical instruments, and in the second video we have seen the structure of a cell, that is what is a human cell, what is its composition, and how the cells communicate with each other. So the cell is a fundamental unit of the body. Okay, the cells communicate with each other through movement of ions. So when the ions move from one place to another place, there will be change in concentration of ions. This change in concentration will change the potential across the membrane. So that potential is called as electric potential or biopotential. So our job is to identify or measure the biopotential generated by our body. The cells communicate with each other through ionic movements. Okay, so each cell will generate some amount of potential. Our job is used to measure the potential. So in this video, you are going to discuss how biopotentials are generated and measured from our human body.
The basic living unit of the body is called as cell. Each organ of our body is a collection of different types of cells held together by intracellular supporting structures. Each type of cell is meant for performing one particular function. The entire body contains above 100 trillion cells. Among these 100 trillion cells, there are 25 trillion red blood cells which transport oxygen from lungs to the tissues. Generally, all the cells have the ability to reproduce new cells whenever the cells of a particular type are destroyed. If the cells feel unfit, the appropriate numbers are replenished further in all areas. Generally, the structure of a cell cannot be viewed by a naked eye. It can be viewed only by biological microscope. The size of a human cell is approximately 5 micrometers. Structure of a human cell. Each cell consists of a centrally located nucleolus called as cell core. The cell core is surrounded by cell body called as cytoplasm. The nucleus is separated from cytoplasm by a membrane called as nuclear membrane. The cytoplasm is separated from the surrounded fluid by cell membranes. The fluid which is present inside the cell is called as fluid, and the fluid which is outside the cell is called as extracellular fluid. There are different types of substances that make up the cell body, and it is called as protoplasm. Protoplasm is composed of water, electrolyte, proteins, lipids, and carbohydrates. So the cell body is made up of protoplasm. Water is the principal fluid present in the cell. It is used as solvent for all chemicals. The concentration of water present in our cell is almost 80%. Next is electrolyte. So the electrolyte is a combination of different types of chemical substances. So the substances which are present in our cells are magnesium, potassium, phosphate, bicarbonate, and elements like calcium, chloride, and sodium. Proteins. the concentration of proteins is almost 10 to 20 percentage. So there are two types of protein structures: structural protein structures, which are long and thin filament like structure. Another protein structure is globular protein, which will provide energy for all the cell functions. The next composition of protoplasm is lipids. Lipids are soluble in fats and solvents. They are insoluble in water. Third substance of protoplasm is carbohydrates. It plays a major role in the nutrient of the cell. It is stored in the cell in the form of glycogen, and they are used to supply energy to the cell. So these are the composition of protoplasm, that is water, electrolyte, proteins, lipids and carbohydrates. So the cell body is called as cytoplasm. It is filled with a fluid called as cytosol, which contains minute granules called as ribosomes, then some particles called as lysosomes, and DNA, which is called as mitochondria. So what are ribosomes? Ribosomes are minute granular particles in the cytosol and are composed of a mixture of ribonucleic acid. Lysosomes. Lysosomes are used in digestive system of the cell. It is used to remove unwanted substances or foreign structures such as bacteria from our body. Mitochondria. It is referred as powerhouse of the cell. It is used to extract energy from nucleus and oxygen. So this is the structure of a human cell. Mechanisms are also used in cell membrane. Okay, so when a cell is in the resting state due to the semi-permeable membrane, there are more number of potassium ions inside the cell and more number of sodium ions outside the cell. So whenever there is a charge imbalance, okay, uh, for example, if you consider a membrane, if equal number of ions are present in inside and outside, that is equal number of positive ions inside and equal number of negative ions outside, they are getting neutralized. But when a cell is in the resting state, you find more number of potassium ions inside the cell and lesser number of potassium ions outside the cell. Similarly, more number of sodium ions outside the cell and lesser number of sodium ions inside the cell. So due to this ionic mismatch, a potential difference exists across the cell structure or the cell membrane. Okay, so there will be two types of potentials generated by our cell membrane. One is called as resting potential, another one is called as action potential. So when cell is in resting state, okay, when the cell is normal, the cell membrane, which is semi-permeable, allows the entry of potassium and chloride ions inside and blocks the movement of sodium ions inside the cell. So due to this, you will find more number of sodium and chloride ions inside the cell body and lesser number of sodium ions inside the cell. Similarly, outside the cell membrane, you will find more number of sodium ions and lesser number of potassium ions. So the concentration of potassium ions inside the cell is 30 to 50 times higher when compared to outside. Similarly, the concentration of sodium ions outside the cell is 10 times higher when compared to the number of sodium ions inside. So this creates a charge imbalance, mismatch in the presence of positive and negative ions. So this charge imbalance creates a potential difference. So a potential difference is developed across the cell membrane due to charge imbalance. This potential difference or the membrane potential is called as resting potential. So when a cell is in the resting state, due to charge imbalance, a potential is created and the potential is called as membrane resting potential. So this potential is caused by different concentration of ions when the cell is in resting state. So when a cell is in the resting state, you will find more number of potassium ions inside and lesser number of potassium ions outside. So the concentration of potassium ions or the charge of potassium ions are negative. So inside the cell you will find more number of negative ions and more number of positive ions outside. Okay, so more number of negative ions present inside the cell, lesser number of negative ions present outside the cell. So the cell is slightly polarized in nature because in its resting state it contains more number of negative ions. So a cell is called as polarized. Just a schematic representation of cell in its resting state. Okay, so when a cell is in the resting state, you can find more number of negative ions inside the cell and lesser number of negative ions outside the cell, or more number of sodium ions outside the cell or lesser number of sodium ions inside the cell. So when the cell is in resting state, it is polarized and more number of negative ions are present. So if you measure the potential difference using a voltmeter, it will read 70 millivolts, minus 70 millivolts as its average value. So it is the resting potential of a cell in a polarized state. Okay, so electrodes can be used to measure the resting potential of a cell. So it is used to read the information. Then using a voltmeter you can read the voltage. So when a cell is in the resting state, the average resting potential ranges from minus 60 millivolts to minus 100 millivolts. And the cell in the resting state is called as polarized cell because it is having more number of negative ions. And the reason for polarization is due to the presence and potassium ions. Now, what is the mathematical representation of this resting potential? So, Nernst equation defines the concentration of resting potential. So, the resting potential calculation is done based on the concentration of potassium ions inside the cell and outside the cell. So, it is based on concentration of or number of potassium ions present outside the cell when compared to the concentration of potassium ions present inside the cell. So this is the equation for the resting potential, which is called as Nernst equation, where Ek is called as the resting potential of the cell. It is governed by R, that is a universal gas constant, T absolute temperature, N valence of the electron, and F Faraday's constant. Ki indicates concentration of potassium ions present inside the cell. K naught is concentration of potassium ions present outside the cell. Another equation for resting potential called as Goldman's equation. So Nernst equation is based on concentration of only potassium ions. Goldman's equation is based on concentration of majority of the ions. That is, the majority ions or the principal ions present in our cell body is sodium, potassium, and chlorine. So Goldman's equation for resting potential depicts all the ions. So that equation is represented here. Concentration of the ions present inside the cell and the concentration of ions present outside. So now when the cell is in the resting state, you will find more number of potassium ions inside and lesser number of potassium ions outside. So due to this charge imbalance, a potential is created, and this potential is called as the resting potential of the cell membrane. Now when a cell is actuated or excited, what will happen? Okay, for example, you take our muscle cells. uh, if you are normal or ideal, your muscle will generate a resting potential of minus 70 millivolts. Now when somebody is pinching you, what will happen? You will feel something. So whenever somebody pinches you, the cells will get excited. Okay, the cells will start working or communicating with each other. So what happens at that time? What will be the chemical composition of the cell? So when a cell is excited, the nature of the cell membrane changes, as it is semi-permeable, and when it is excited, the cell membrane allows the entry of sodium ions inside the cell. At the same time, it allows the movement of potassium ions outside the cell. So the nature of the cell is they always move from higher concentration region to lower concentration region. So when the cell is in the resting state, you will find more number of potassium ions inside and more number of sodium ions outside. So when a cell is excited, this nature of semi-permeable membrane changes. So more number of potassium ions from inside will flow outside the cell membrane. Similarly, sodium ions which are present outside the cell will move towards inside the cell. So there is a change in the ionic concentration. So whenever the ionic concentration changes, the potential will also change. Okay, now you will find more number of sodium ions inside the cell and more number of potassium ions outside the cell. So we are measuring the potential with respect to inside the cell. So when a cell is excited, you can find more number of potassium ions inside and more number of potassium ions outside. So there is a change in the polarity. Now if you measure the potential will be a positive value because you find more number of sodium ions inside the cell and more number of potassium outside. So you will find a positive value. Okay, so this is a diagram for the representation of action potential. So when a cell is excited, potassium ions moves outside and sodium ions which are present outside the cell will try to enter into the cell. Okay, so due to this, you will find more number of potassium outside the cell and more number of sodiums inside the cell. So this is a schematic representation of a measurement of the action potential. So when a cell is excited, the charge composition changes. More number of positive charges inside the cell due to sodium ions and lesser number of sodium ions outside the cell. So due to this charge imbalance, a potential is created. So this potential called as action potential. So this potential is called as action potential because the cell is excited. An action is done by the human. Okay, so cell is excited by some action. So The potential which is generated by the action of a cell is called as action potential, and its average value is 20 millivolts. Okay, so when the cell is in the resting state, it is in polarized form. Okay, when you excite a cell, the charge composition changes. So it is called as depolarization. So when a cell is excited, the potassium ions move inside the cell. Sorry, the potassium ions inside the cell moves outside, and the sodium ions which are present outside the cell moves inside. So they will not remain there for a longer period. So after that, they will become normal. So what will happen is the sodium ions which have gone inside the cell will come outside. Similarly, the potassium ions which is present outside the cell will come inside the cell. So this is called as repolarization. From excitation, the cell will come to normal state. So initially in the resting state, a cell is in the polarized state. Then due to excitation or by some action, it becomes depolarized, and after depolarization, after some time, the cell will again get repolarized. That is, it will have more number of potassium ions inside and more number of sodium ions outside. So again, if you read the voltage, the average value will be minus 70 millivolts. So it is called as cell repolarization. So how the structure of the membrane potential will look like? So initially when the cell is in the resting state, you will read the resting potential or membrane potential's average value as minus 70 millivolts. So it is minus because we are measuring the potential with respect to inside the cell. So inside, more number of potassium ions are present when the cell is in the resting state. So you will read minus 70 millivolts. Then when you excite a cell, the concentration of the ions changes. Potassium ions move outside, sodium ions move inside. So due to this, the potential also changes. Now when you measure the voltage, instead of a negative value you will read a positive value because the charge of sodium ions are positive. So from minus 70 millivolts it will read it as an average of 20 or 30 millivolts. So this is called as action potential. So the resting potential is minus 70 millivolts, the action potential is plus 20 millivolts. Then after some time, again the charges will move back to their original position. The potassium ions will move from outside the cell membrane to inside the cell membrane. Similarly, the sodium ions will flow out of the cell membrane. So it is called as repolarization. So initially the cell is in polarized state, then due to excitation depolarization occurs, then after some time again the cell will get repolarized and it will come to its normal form, that is resting form, and the potential, if you read it, it will indicate minus 70 millivolts. So how this action potential moves? Okay, so this resting potential or action potential is generated by a single cell. Okay, so now how the biosignals are generated? Okay, so biosignals or the electrical signals are generated from our body due to the movement of ions inside our cell. So when cell communicates with each other, the ions move from one place to another. That is, the ions move from inside the cell to outside the cell or outside to inside. So this communication between the adjacent cells gives rise to propagation of action potential from one cell to another cell. So the job of a medical engineer is to measure the concentration of resting and action potential present in our body. Okay, so only for that we are designing the equipments. Okay, so the input is from human body, it is taken to a system for processing, and it is fed to an another device as a display, or it is recorded. Okay, so in simple terms, human physiology is the study of body and its function in each of the different systems in a living body. So this indicates that our human body contains different types of organs, and the job of biomedical engineer is to understand the functionality of each organ and analyze them and design equipments for diagnosis and therapeutic purposes. So what are the different types of physiological systems present in our body? So all of us know the answer for this, starting from vision, that is eyes, hearing, ears, smell, nose, taste, tongue, inspired air, nose, okay, muscles, speech. So all these systems require or have specialized cells. So we have to develop systems such that we are able to sense the signals produced by those organs or the organ cells, process them, and give it to doctors for diagnostic and therapeutic purposes. So the different physiological systems of human body: skeletal system, muscular system, nervous system, endocrine system, cardiovascular system, respiratory system, digestive system, urinary system, reproductive system, integumentary system, lymphatic and immune system. So all these systems require specialized instruments because all these organs are different from each other. Every organ produces different types of biosignals. So we are in a state to read these signals, process them, and take it for the doctor for treatment. Okay, so based on this, we classify the different types of signals produced from our human body. Okay, so what are the different bioelectric signals produced by the various organs of human body? So the signals which are produced by our eyes, biosignals generated by organs or the cells present inside our eye, is called as electrooculogram. The biosignals produced by our heart or cardiovascular system is called as electrocardiogram. The bioelectric signals produced by our muscular structure is called as electromyogram. Pulse rate cardiovascular system, phonocardiogram produced by heart. Blood pressure, again a signal produced by cardiovascular system. Blood flow, cardiovascular system. So like this gram indicates a name of the biosignal. Okay, then if you mention something called as electrocardiograph, then the graph indicates the instrument. Gram indicates the signal name and the graph indicates the instrument